After Part 1 introduction, I believe now you are much better aware of the anatomy of intestinal mucosal immunity, food allergy animal model used in this study, and what is known about type 2 allergy. Now we're going to look into this article and ask how does this article add value to the knowns, and what are the caveats for enriching low abundance immune cells. Then we are going Part 2 Article Presentation Transcriptional Atlas of Intestinal Immune Cells reveals that neuropeptide alpha CGRP modulates group 2 innate lymphoid cell response. Immune cell profiling in LP and PP. So, the authors systematically dissect the cell types from myeloid lineage and lymphoid lineage, and compare cell abundance in LP lamina propria and PP Peyer's patch. This provides a clear overview of cell type enrichment in each site. Let's take a close look at what is enriched in LP. Notably ILC2 and mast cell. ILC2 and mast cells are significantly enriched in LP, and also plasma cell is significantly enriched in LP. In contrast, resting B cells and germinal center dark zone and light zone B cells are significantly enriched in Peyer's patch. And you can see on the right-hand side, this staining also indicates that IgD stands for a marker for immature B cell. Most of them are located in Peyer's patch, while CD1A3 stands for marker of plasma cell. Predominant Peyer's patch is a structured lymphoid tissue with germinal center, as you can see in the left-hand side. Within Peyer's patch, there is germinal center, and within germinal center, there are dark zone and light zone. Within the dark zone, where naive cell migrate into the dark zone and undergoes clonal expansion followed by somatic hypermutation, and then the cell migrates into the light zone where selection and class switching take place, and then B cells mature into plasma cell and enriching low abundance cell types. To enrich low abundance cell types, they also employ two strategies. One is to exclude all immature B cell using the marker IgD and select IgD low cell. The other strategy is excluding T cells CD3 positive and mature B cells CD19 positive, and selecting double negative cells. You might wonder what's the purpose of this enrichment process, as you know that Peyer's patch is a secondary lymphoid organ enriched with T cell and B cell. It would be difficult to detect minor cell types under such condition, and so the authors use flow cytometry gating strategy to exclude immature B cell or both T and B cell in order to enrich low abundance myeloid lineage. Immune cell type profiling in type 2 food allergy. Most low abundance cell types become readily enriched in CD3 and CD19 double negative cell population. As you can see here, ILCs and myeloid lineage cells are now readily enriched and become much more apparent to be observed. ILC2 and mast cells are significantly increased in OVA groups compared to control PBS group, as you can see here. And there are 27 cell types are systematically analyzed. The most enriched cell are plasma cell. and resting CD4 T cell. However, there's no significant differences between two treatment groups. So in OVA-induced type 2 food allergy mouse model, we can see that mast cells and ILC2s are significantly upregulated in OVA-induced type 2 food allergy mouse model. Transcriptional atlas of intestinal mucosa. To create a transcriptional atlas of intestinal mucosa, firstly, the mouse are subjected to OVA sensitization and oral challenge. Following that, LP and PP are taken from enzymatic digestion to isolate immune cells therein. The immune cells are then stained with cell type specific marker, immune cell sorting, and subjected to single-cell RNA-seq. The lower left shows the transcriptional atlas of each cell types in PP and LP. The right-hand side are the sum of subpopulations categorized in using common names like B cell, T cell, myeloid cell. There are subpopulations within. It is apparently that PP has enriched single-cell RNA-seq and transcriptome-based cell type classification. Immune cells are either directly used for single-cell RNA-seq or first stained and sorted by flow cytometry followed by single-cell RNA-seq. And then each cell are first barcoded by barcoded primer beads, which later barcode the cDNAs, reverse transcribed from the RNA of each single cell. The cDNAs are then sequenced and analyzed for cell classification. The cell classification was mostly consistent with cell ILC2 and mast cells show the most prominent alterations in their transcriptome upon OVA challenge. ILC2 and mast cells are those cell types with greatest alteration in their transcriptome profile as seen in the rightmost figure. As you can see, the darker the color indicates the more dysregulated expression gene. And ILC2 and mast cell have the most prominent differentially expressed genes upon OVA challenge. And in the middle, you can also see a shift in OVA group versus PBS group in cell type 12 and 26. Cell type 12 indicates distribution of each dot here represent the transcriptome variation between single cell. And the color indicates the expression abundance of each topic. And topic means a series of genes whose expression pattern is consistent within a specific cell population. And gene weight means relative expression level within single gene. And as you can see here in topic 2, OVA group gene weight cumulative curve, the red line shows, is most distinguishable throughout all 10 topics, as you can see here. And calcitonin is the most abundantly expressed gene in topic 2, as you can see in the lower left panel. All these data supports that calca is the top gene in ICL2. Calca stands for calcitonin-related polypeptide, alpha, and its gene product is called alpha-CGRP. So in the left panels, You can see here alpha CGRP when compared with two other genes. Alpha CGRP significantly increased upon OVA challenge, while the other two genes is significantly decreased upon OVA challenge. And now in the right panel, we can see that comparing across all different cell types, calca is specifically expressed in ICL2 cell. And when compared with these two other genes, its expression abundance is. Now that we know alpha CGRP expression in ICL2 is induced by OVA challenge, so what's the function of such induction of alpha CGRP expression? Now the author used in vivo primary culture of ILC2 to answer this question, and then we also found that IL-25 is capable of eliciting prominent transcriptome induction of a series of genes, and such induction can be reversed when co-treated with alpha CGRP. It means that alpha CGRP reverses the transcriptional activation elicited by IL-25, a known cytokine initiating type 2 inflammation. So besides inhibiting the gene induction of ICL2, alpha CGRP also have some effects on type 2 cytokine expression on ILC2. Let's take a look. The IL-13 level is increased upon the IL-25 treatment, and with co-treatment with alpha CGRP, IL-13 expression is reduced, while that of IL-5 is increased. So this is summarized in here. Alpha CGRP selectively inhibits IL-13 but not IL-5. And on ILC2 expression, as we can see here, IL-25 alone is capable of increased ILC2 expansion. However, such expansion is reversed when co-treated with alpha CGRP. So the dividing cell. So to know what's the function of alpha CGRP expression in vivo under type 2 inflammation, the author used daily intraperitoneal injection of either only IL-25 or IL-25 plus alpha CGRP. And in this staining, they also use KLRG1 to stain the ILC2 and CD3 to stain T cell. As we can see here, in IL-25 alone can induce the green cell number. The green cell is ILC2. And in co-treatment group, the green cell number is significantly reduced compared with IL-25 alone. And the both staining picture and quantification results in the right panel shows consistent trend of significant decrease of ILC2 number in co-treatment group compared with IL-25 alone. To further understand what's the function of enhanced alpha CGRP expression during type 2 inflammation, the author goes further to use the OVA food allergy model. And also the alpha CGRP this time is using oral gavage, intragastric challenge rather than intraperitoneal injection. And as you can see in the lower two panels, alpha CGRP oral co-administration can suppress OVA-induced ICL2 number in lamina propria, and also reduces the OVA-induced mast cell number in. So, to know what's the function of alpha CGRP under normal physiological condition, we also use tissue immunostaining to look at the colocalization of the CGRP and acetylcholinergic neurons. And surprisingly, we found that there's a perfect match of this colocalization, suggesting that alpha-CGRP is produced by acetylcholinergic enteric neurons. We also further wonders whether in the absence of alpha-CGRP what will happen. So they use genetic perturbation in alpha-CGRP gene, and we found that in KLRG1 stands green. The green cells significantly increased in this genetic perturbated alpha-CGRP gene. And inconsistent with the staining data, the quantification results shows that delta exon 5 calca group has significantly increased the number of ILC2, suggesting that under steady states, alpha-CGRP is produced by acetylcholinergic enteric neuron and controls KLRG1 positive ILC2 cell number during homeostasis in vivo. And surprisingly, we also found that the DCLK1, which stands for tough cell. Tough cell is the chemosensory cell, which produces IL-25 to initiate type 2 responses. And in the genetic perturbated calca gene, tough cell stance also increased and inconsistent with the staining results. The quantification also shows that in the absence of calca expression, tough cell number also increased significantly. So alpha-CGRP not only controls the homeostasis of ILC2 cell, but also tough cell. So to know what happens when alpha-CGRP binds to plasma membrane ILC2 and what's the signal transduction events there, the author used in vitro to culture primary ILC2 obtained from lamina propria, and upon treatment of alpha-CGRP, the cells are then collected and subjected to chromatin accessibility analysis, and those loci with upregulated chromatin accessibility are then subjected to pathway analysis. As here we can see that the pathway adenylate cyclase activity has been significantly increased. To know what happens during alpha-CGRP treatment, they also then measured cyclic AMP level after 30 minutes of alpha-CGRP treatment. As you can see in the right panel, cyclic AMP intracellular level has increased significantly after 30 minutes of treatment. Knowing that alpha-CGRP treatment can increase the intracellular cyclic AMP level in ILC2, but what happens when intracellular cyclic AMP level has increased? And how does cyclic AMP influence the ILC2 expansion? Here the author used a small molecule called forskolin. Forskolin can stimulate adenylate cyclase activity, thereby increase intracellular cyclic AMP level. And then we'll see whether it can mimic the inhibitory effect of alpha-CGRP in suppressing ILC2 expression. In the left-hand side panel, you can see indeed, upon treatment of forskolin, the population of expanded cell has significantly decreased, and as here shown in the flow data and also in the quantification data, shows that small molecule forskolin, which can stimulate the increase of cyclic AMP level, indeed mimic the inhibitory effect of alpha-CGRP and suppresses the ILC2 expansion. On the other hand, the author also used adenylate cyclase inhibitor SQ22 and 536, and they have observed that this can partially reverse the inhibitory effect of alpha-CGRP on ILC2 expansion. Now let's go over the graphic summaries of the entire article and refresh the significance of this article. So first, the author has successfully established an OVA-induced food allergy model where they use this animal and collect immune cells from their participation and lamina propria, make them subjected to single-cell RNA-seq, and then did computational analysis and topic modeling, where they found that alpha-CGRP is the most significant upregulate gene in ILC2 in response to OVA stimulation. And also during homeostasis, it is the cholinergic enteric neuron that secretes alpha-CGRP to control the cell number of ILC2. And in ILC2, the plasma membrane has G protein coupled receptor that can respond to alpha-CGRP, and through this G protein coupled receptor, they controls the cyclic AMP level, where the cyclic AMP response genes then control the expression of IL-5 and inhibits the cell expansion of IL. And the significance of this article is that the authors has established a transcriptional atlas of intestinal immune cell profile at different status during food allergy, and they discovered that neuropeptide alpha-CGRP involves in modulating ILC2 responses, both the cytokine expression level and ILC2 cell expansion. And so these findings suggest that alpha-CGRP could be a potential therapy for ILC2-mediated food allergy. During reading this article, I raised several questions and looked for the answer. Here I'd like to share with you my questions and the ideas inspired by this article. So my first question is whether suppression of ILC2 expansion can alleviate type 2 allergic symptoms remain to be determined in human population, because ILC2 is only account for one of the many features of type 2 allergy. By checking literature, I found that in mouse model of ILC2 deficient mice, type 2 inflammation is severely impaired, and this results strongly support that almost all cell types involved, ILC2 is the most critical upstream reactor and effector. Actually, it plays the initiating role in type 2 inflammation. That's why ILC2 is so important in type 2 response. My second question is that what is the mechanism conferred by alpha-CGRP to suppress both ILC2 and mast cell at the same time? Is there a hierarchical relationship between ILC2 and mast cell? Yes, indeed, similar to my previous question, because ILC2 plays an initiating role. It secretes type 2 cytokines, including IL-5 and IL-13, which can promote IgE antibody production and then exacerbate mast cell-mediated inflammation. So it is true that there is a hierarchical relationship between ILC2 and mast cell. So the ideas inspired by this article is that based on these knowledge, I think antibody designed against ILC2 marker can eliminate ILC2 population and thereby confers resistance to ILC2 response. The other idea is that the PK property design for alpha-CGRP can minimize its chance to cross blood-brain barrier that cause side effects in CNS migraine. An oral alpha-CGRP capsule dissolved in intestine could be a promising prevention and therapy for food allergy. So the authors have now established the correlation between human allergy and circulation ILC2. I checked this on clinicaltrials.com. So now with that, I'd like to thank you for your attention, and let's look forward to a better therapy to control type 2 food